Today, I am taking up the topic photosynthesis. Photosynthesis is a very big topic, so it is divided into three parts. Today, I am going to talk about first part of photosynthesis. It includes earlier experiments, structure of a chloroplast, photopigments and solar spectrum. Photosynthesis is one of the important processes occurring in the nature. Green plants are the only organisms that can trap solar energy or the radiant energy coming from the sun. Directly or indirectly, all the living beings depend on the solar energy for their survival. The sun releases vast amounts of radiant energy, but only 0.2% of this radiant energy is used by the plants in photosynthesis. So plants, by using sunlight, water, carbon dioxide and photopigments or chlorophyll, synthesize glucose. The first product of photosynthesis is glucose. From glucose, all other types of carbohydrates, proteins and fats are synthesized. But the basic product is glucose. And all the herbivores, that is mostly animals, they cannot synthesize their own food. So all the herbivores or all the animals invariably depend on the plants for their food needs. Either they depend directly as herbivores or indirectly as carnivores. Then, photosynthesis is an anabolic process where energy is trapped. It is an anabolic process. Energy is trapped into glucose. Then, the overall equation of photosynthesis can be described like this. 6 CO2, that means 6 molecules of carbon dioxide, 12 molecules of water. They combine in the presence of sunlight, and with the help of the chlorophyll, they produce glucose, 6 molecules of water and 6 molecules of oxygen. Remember, all the oxygen present in the atmosphere comes only through photosynthesis. Without photosynthesis, there will be no oxygen. So, photosynthesis not only helps in synthesizing food, but also it contributes to the atmospheric oxygen. And plants are the only structures or organisms which trap carbon dioxide. Otherwise, the carbon dioxide levels build up in the atmosphere, and now we are experiencing the bad effects of carbon dioxide in the form of global warming. We will be reading or studying more and more about the ill effects of carbon dioxide in the chapter pollution. Then, in photosynthesis, what is happening? The solar energy, which is a radiant energy, is converted into chemical energy, that is glucose. Then, early experiments. We know Aristotle is the father of zoology. Theophrastus is the father of botany. They were ancient Greek philosophers. They believed that plants obtain nourishment from soil. They were not aware of chlorophyll or other things. Then Helmont's experiment. In 1648, Helmont conducted one wonderful experiment. Based on, it is really a scientific experiment. He weighed completely dried soil of 200 pounds. First he dried the soil completely and weighed it. He took 200 pounds, he had placed in a big pot. Then he planted in that pot a willow plant weighing only about 2 pounds. 5 pounds. Weighing about 5 pounds. Then he added rainwater to the soil and covered the pot with a perforated iron plate so that dust cannot fall into the pot and weigh and increase its weight, increase the weight of the soil. Then he kept it in the ground and he maintained the plant for 5 years. During these 5 years, he went on adding only rainwater or distilled water to the plant. After 5 years, when he weighed the plant, it weighed 164 pounds. So originally the plant weighed only 5 pounds. After 5 years, its weight increased to 164 pounds. Then he took out the soil from the pot, dried it completely and weighed. When he weighed what he found, the initial input in the pot was 200 pounds of the soil. But he found that these 200 pounds became only 198 pounds. That means the soil has lost only 2 pounds. Then how to explain this gain? He had no answer. You could understand. He thought that the weight was gained from substances absorbed from water. That is Helmholtz's experiment. Then Joseph Priestley, he conducted one very interesting experiment in 1772. Joseph Priestley was born in England and he discovered a number of gases. Among them, hydrogen, oxygen, laughing gas, N2S, and many more. One day, he conducted a simple experiment. He took a candle and placed it in a closed chamber. The candle continued to burn for some time, then it went off. Then what he did? In another experiment, he placed a mouse in a closed chamber. The mouse lived for some time and then it died. Then he assumed that the burning candle or the living mouse injured the air. Here, injured the air means absorbed something from the air or removed something from the air. And that we know now is oxygen. Then what he did? He placed a plant. A small plant in the pot along with the candle. In a closed chamber, he placed a burning candle and a plant. Then, to his surprise, the candle continued to burn. Then he repeated the experiment with the mouse. When he placed a living mouse and a plant in a closed chamber, the mouse continued to live indefinitely. So from these experiments, what he inferred was that whatever the gas removed from the air by a burning candle or a living mouse is replenished by a green plant. That means whatever is lost from the air is incorporated by the green plants. Now we know that it is nothing but oxygen. That is about the experiment of Joseph Priestley. Then comes John Ingenhouse's discovery in 1778. He did another interesting experiment. He placed a green aquatic plant in the sunlight. He placed the green aquatic plant in a sunlight and found that small bubbles were evolving or coming or forming around the green substance, that is chlorophyll. From this experiment, what he showed was sunlight is necessary for photosynthesis. If the Priestley's experiment, if it is conducted in the absence of sunlight, again the candle will stop burning after some time, the mouse will die after some time. But the experiment will be success only if the apparatus is placed in sunlight. So Indian house discovered that the green parts of the plant produce small air bubbles or some bubbles, gas bubbles around them. And these bubbles are produced only in the presence of sunlight. So in that way, he has shown that oxygen is released during photosynthesis. Then, Jean Senebier's erroneous experiment. He proposed one theory. I have told you, during photosynthesis, oxygen is released into the atmosphere. Then the question arises, from which component the oxygen is released? Is it coming out from the carbon dioxide or from water? Both carbon dioxide and water are used in photosynthesis, and both of them have oxygen. Then which one of these two oxygen is released? Senebier proposed that during photosynthesis, carbon dioxide is split into carbon and oxygen. Carbon is used in the synthesis of carbohydrates and oxygen is released. That was his theory. But this theory was proved to be wrong by an ingenious experiment conducted by Cornelius van Niels. It's really very interesting. Green plants, what they do? I have told you they use 4 things in photosynthesis: sunlight, carbon dioxide, water, and chlorophyll. There are some bacteria which do not depend on water for their photosynthesis. So here, what he did, he raised some bacteria known as sulfur bacteria. Sulfur bacteria. Sulfur bacteria use H2S instead of water. So when this bacteria, the sulfur bacteria was studied, he found that instead of oxygen, sulfur gas is released. Sulfur gas. And the sulfur gas obviously comes from H2S, hydrogen sulfide. So from this experiment, he has proven that oxygen, that means, here one more point I want to add, green plants, what they use H2O instead of H2S. That means, when in the sulfur bacteria, sulfur gas is released from H2S, he concluded that in photosynthetic plants, oxygen is released from water. That is what he found out. And now we know that Niels' experiment is correct. He correctly predicted that oxygen evolves from water and not from carbon dioxide. The findings of Van Niels was supported by the experiments of Robert Hill. Then, still there was some doubt whether oxygen is released from carbon dioxide or from water. The clinching evidence that oxygen comes only from water is obtained from the experiments of Samuel, Rubin, and others. Here what they did, they used heavy oxygen, that is 18O instead of normal oxygen, 16O, in water. And they have found that the oxygen released during photosynthesis is 18O and not 16O. That means in the carbon dioxide, 16O is present in the experiment, and in water, 18O is present. The oxygen released was 18O. It clearly proves that oxygen comes from splitting of water, or there is a term for this. It's called photolysis of water. That means water is split into hydrogen and oxygen in the presence of sunlight. So in this way, now we know that oxygen evolves from water and not from carbon dioxide. Now, for photosynthesis, all these experiments prove that these four are required, that is chlorophyll, water, carbon dioxide and sunlight. These experiments can be done in a simple way also. If you cover a green leaf with a black paper, you find that the covered part of the leaf fails to synthesize glucose or starch. That is because of the absence of sunlight. If you place a part of the leaf in a test tube or a container containing KOH, what happens? KOH absorbs all carbon dioxide. So carbon dioxide is not made available for photosynthesis, and that part of the leaf inserted into the container having KOH fails to synthesize starch. Like that, you can show that all these four requirements are needed for photosynthesis. Then coming to the structure of chloroplasts. Chloroplasts are present in all the green plants. You know that chloroplasts and mitochondria, both of them were once free-living bacteria. Still they exhibit a number of bacterial characters. Somehow they got incorporated into the eukaryotes and they became symbionts first, and then they became a part and parcel of the cell, or they become cell components. The mitochondria and the chloroplasts have many similarities. Both of them are made of double membranes. This is called outer membrane, this is the membrane. In mitochondria also we find two membranes. Then, in both chloroplasts and mitochondria we find circular DNA molecules. Double-stranded circular DNA molecules. Then we also come across ribosomes. These ribosomes are 70S type. In eukaryotes, we find 80S ribosomes. But in mitochondria and chloroplasts, we find 70S ribosomes, just like bacteria. Because bacteria also have only 70S ribosomes. So all these factors show that they have great resemblance to bacteria, because they were the descendants of bacteria. Then coming to the chloroplast. This is the chloroplast, a single chloroplast. This is a double membrane, outer and inner membranes. The chloroplast contains stroma. The fluid present in the chloroplast is called stroma. Inside the stroma we find membranous structures present in the form of grana. These grana are made of thylakoid membranes. Thylakoid membranes are closed sac-like structures. Closed structures, sac-like structures, and they are arranged one above other like this. The arrangement of these are like the arrangement of coins. So one pile of coins is called grana. Single one is called granum. So this is one granum, this is one grana, this is one grana, this is one grana. Different grana are connected by stromal lamellae. Stromal lamellae. So this is one stromal lamella, this one stromal lamella, like that. Then the chlorophyll is actually present in the thylakoid membranes of grana. Is present in the thylakoid membranes of grana. I will be talking more about the thylakoid membrane after just a few minutes. Then, in the stroma, we come across lipid droplets and also starch granules. These are derived from the glucose synthesized through photosynthesis. And in photosynthesis, there are two important reactions which are known as light reaction and dark reaction. These names are not so correct. Light reaction means light dependent reaction. Dark reaction means light independent reaction. So let there be no confusion. Light reaction occurs in grana, or light dependent reactions occur in grana or granule membranes, whereas the dark reactions occur in stroma. All the enzymes required for dark reaction are found only in the stroma. The dark reaction also requires a lot of energy, and this energy comes from ATP and NADPH. They are available in stroma. And interestingly, ATP and NADPH are synthesized in the granule membranes or the thylakoid membranes. We'll be talking more about these things when you take up the light reactions and dark reactions. Here, if this is one granum, I have magnified a small part of the membrane here. Thylakoid membrane. A small part of thylakoid membrane. We'll be knowing more about these things in the next class when you take up the light reaction. Just for the time being, remember, in the thylakoid membranes, we find photosystem 2, photosystem 1. So the photosystems 1 and 2, which are necessary for photosynthesis, are present in the thylakoid membranes. The sunlight actually stimulates these photosystems 1 and 2. Then, in the thylakoid membranes, we also come across plastoquinoline, one very important substance which connects both electrons and hydrogen ions. Then, there are electron transport systems or chains. This is one, this is one. Through these chains, electrons are transported. How that happens, we will study in the next class. Just for that, remember, in the thylakoid membranes, we come across the photosystem 2 and 1, electron transport chains or systems, and also one enzyme known as ATP synthetase. This one. Through ATP synthetase, when hydrogen ions travel like this, ATP synthesized. Then, this is thylakoid lumen. That means this one. This is outside stroma. So this is stroma. This is the lumen of thylakoid. Then in the sunlight, water also undergoes photolysis to produce electrons, hydrogen ions and oxygen. This process also occurs in the thylakoid lumen here inside. We'll be studying these things more clearly in the next class, but for the time being, don't get confused. Then, photopigments. I have told you, photopigments are present in the thylakoid membranes. These photopigments are 4 types. Chlorophyll A or alpha, chlorophyll B or beta, xanthophylls and carotenoids. Not only their color, their reactions are also different. Chlorophyll A, one more point I have to explain. In the sunlight, we find 2 spectra. Visible spectra, invisible spectra. Visible spectra means VBGR. Violet, indigo, blue, green, yellow, orange and red. These 7 colors form the visible spectrum. Before and beyond this visible spectrum, there is invisible light, that is ultraviolet light, infrared rays, radio waves, X-rays, like that. There are so many invisible radiation. Then, during photosynthesis, mainly the blue light and red light are applied, or blue spectrum and red spectrum are employed. So I have given the wavelengths here. The blue spectrum has a wavelength from 430 to 470 nanometers. The red light has 660 to 760 nanometer wavelength. Others I have not drawn because they are not those so important. So for photosynthesis, blue light and red light are very important. Other lights are not so important. They are also employed but not so important. Here I have drawn two lines. I simplified what's given in the textbook. Rate of photosynthesis. It is very high in the blue spectrum and also in the red spectrum. Then coming to the absorption of the spectrum, again in the blue range it is high, and it is high again in the red spectrum range. So, in the photosynthesis, the blue light and the red light are mainly employed. Now let us see. Chlorophyll A absorbs mainly red and blue light. So it is very, very important. Chlorophyll B, it is yellow-green in color. Xanthophylls, yellow. Carotenoids, yellow and yellow-orange. In this way, there are 4 types of photopigments. Chlorophyll A, chlorophyll B, xanthophylls and carotenoids. So, chlorophyll A absorbs mainly red and blue light. Now, a question arises. Why the leaves appear green in color? The leaves reflect green light. They absorb red and blue light but reflect green light. That's why leaves appear green in color or greenish in color. Chlorophyll B, xanthophylls and carotenoids, they absorb light in other spectrums other than blue and red, and that is transferred to again to chlorophyll A. So even the light trapped by these pigments is transferred to chlorophyll A. So ultimately, chlorophyll A participates in photosynthesis. Then these 3 pigments have also other function. They prevent photooxidation. They prevent photooxidation. Just remember the term photooxidation. I will be talking more about photooxidation in the next class. So this is about the earlier experiments, structure of a chloroplast, photopigments and solar spectrum. With this knowledge, let us study the next topic, light reaction in plants. Thank you.